Good afternoon, everyone. This is going to be a continuation of the biochem notes. We have the test on Friday this upcoming week, and this first chapter is the electron transport chain and the ATP synthase. I'm not going to be doing separate videos for the reactions this time because they're kind of straightforward. They're also in the lecture slides. These are just going to be the highlighted portions from when the professor went over them in class. Okay, so let's jump into oxidative phosphorylation. So energy from reduced fuels is used to synthesize ATP in animals. You're going to be using carbs, then lipids, and then amino acids. And these are going to be the reduced fuels. Again, the electrons from these fuels are going to be carried, and they're going to be used with these reduced cofactors NADH or FADH2. And therefore, this energy is what drives ATP synthesis. The process of ATP being formed from ADP and PI, it's very thermodynamically unfavorable. So you need to actually couple it with something that will generate some energy. So therefore, we take the flow of H plus downstream as the force to cause this phosphorylation of ADP. And then the energy released by the electron transport chain, it pumps out these hydrogens against the gradient. So what do we mean by against the gradient? So we're going from an area of low concentration of protons to the outside the inner mitochondrial membrane, which has a large amount of positives or a large amount of protons. So we're going from low to high instead of high to low. That's through the ETC, but however, we also have the flow of protons downstream through the ATP synthase, and that's what's going to help us do the phosphorylation. Okay, we were just talking about this, the proton gradient. It's only on a membrane that is impermeable for ions. The plasma membrane in bacteria would be an example. However, the inner mitochondrial membrane is an example for eukaryotes concerning animals. Okay, so the electron transport chains complexes are going to have this downhill electron flow, and again, they're going to be pumping out the hydrogens. Some of them pump out hydrogens, some of them don't. Complex 2 does not pump out any hydrogens. This downhill entrance of hydrogens back into the mitochondrial matrix, so this time, once we've pumped out a lot of hydrogens into the membrane, we're going to go downhill now through the ATP synthase, and this is where we're going to actually make the ATP. We have this chemiosmotic model, and again, energy is made by the electrons going through the electron transport chain to the matrix. You pump out hydrogens through those complexes in the inner mitochondrial membrane, and then once you get a lot of hydrogens into that area, the inner mitochondrial membrane where you have a ton of protons, they're going to go through the ATP synthase back into the matrix. Remember, in this electron transport chain specifically, this pair of electrons, these electrons are going through the complexes. They're going to be ultimately taken up by oxygen, and you're going to reform water. Chemiosmotic model, let's just draw it. Why not? Okay, so outside, this is the inner mitochondrial membrane. On the outside, it has a ton of hydrogens. We're going to have this electron carrier, and we have these four complexes. The electron is going to go through the complexes of the inner mitochondrial membrane, and some of these, 3 out of 4 of those complexes, are going to pump out some hydrogens. This electron, when it's finished going through the electron transport chain, it's going to go mix with O2, and it's actually going to just form H2O. Oxygen, again, is the final electron acceptor. And then we have ATP synthase, and these hydrogens that were on the outside in the inner mitochondrial membrane, remember that's a high area of positives. Those hydrogens are going to flow back into the matrix, and when we have that downhill flow of the hydrogens, that's going to drive ATP synthesis. Okay, let's talk about the membranes of the mitochondria. We have the outer membrane, which is permeable using these porins. And then we have the inner membrane, which is impermeable to most small molecules and ions. You need the specific transporters for any type of transport when you're talking about the inner mitochondrial membrane. Let's talk about the mitochondrial matrix now. So, we have things such as the pyruvate dehydrogenase complex, where we're taking pyruvate to ACOA or acetyl-CoA. We have other reactions too. We have the Krebs cycle enzymes, where we're making 3-NADH1, FADH2, and 1-ATP. We also have beta oxidation enzymes here in the mitochondrial matrix. That's going to make 1 ACOA and 1/2 NADH and 1/2 FADH2. Actually, I think that's incorrect. I need to change that. The beta oxidation one is incorrect. I need to go back and change that. Just disregard that. Separates. So the mitochondrial matrix will separate cytosolic and matrix pathways. The cristae are going to be convolutions, like these folds. That's what I mean by convolutions, in the inner membrane of the mitochondria. And mitochondria of cells with high metabolic activity have more cristae. You need that surface area. Okay, let's talk about the respiratory chain. This is the series of electron carriers. The dehydrogenases will collect electrons, and they're going to be funneled into universal electron acceptors, such as the nicotinamide nucleotide shuttles, which are NADH, NADPH, or you're going to also have flavin nucleotides, which include FAD or FNN. Okay, nicotinamide nucleotide-linked dehydrogenases are going to be your NAD or NADP. So NAD is going to be the reduced form. Actually, this is actually reversed. Yeah, this is reversed. I just noticed something in this slide. There's something incorrect in this slide of the professor. Let me go back. So NADH, this is reduced. However, when we have NAD, this is oxidized. In the slides, this is the other way around, but this is correct. NADH is reduced. The NAD plus is oxidized. 2 hydrogen atoms are going to be removed from the substrates in the form of 2 electrons. One is transferred as a hydride, which is H- with 2 electrons, and this is going to be transferred to NADP, and then the other one is H+. We didn't need to know that information, but accidentally went through it. Okay, let's go to the reactions where you could get some NADH. You could have it from the Krebs cycle, the formation of acetyl-CoA glycolysis, or beta oxidation. An example of a reaction that's catalyzed by NADP or NAD dehydrogenases would be the alpha-ketoglutarate becoming succinyl-CoA, malate becoming oxaloacetate, or glucose 6-phosphate becoming the lactone in the pentose phosphate pathway. This one has NADPH. Okay, let's talk about the flavoproteins. Flavoproteins are tightly, covalently bound flavin nucleotides, and this will accept either 1 electron. So if it's accepting 1 electron, this is where we get the semiquinone. And then if it's selecting 2 electrons, for purposes of the class, she wanted us to just focus that it's accepting 2 electrons. This is where we get something like FADH2. Okay. And we get this from beta oxidation, the citric acid cycle, or again, pyruvate becoming ACOA. That's where this FAD is involved. What is actually transferred? So, 3 types of electron transfers occur in oxidative phosphorylation. You have electrons. You could also be moving them as hydrogen atoms or hydride. There are 3 other types of electron-carrying molecules in the respiratory chain. One of the first new ones is ubiquinone. This is a lipid-soluble long-chain hydrocarbon. It's a benzoquinone, and it can accept 1 or 2 electrons. It's located in the inner mitochondrial membrane. So when it's accepting 1 electron, this is called ubisemiquinone. But if it's carrying 2 electrons, this is known as ubiquinol, as in, ubiquinone, but instead of O-N-E at the end, it's just O-L, so ubiquinol. This moves in this inner mitochondrial membrane. It's actually mobile. We also have cytochromes. There's cytochrome A, B, and C. We're only for the purposes of the class, the professor wanted us to focus on cytochrome C. They have this iron heme group, and it's a prosthetic group as well. They carry only 1 electron, and the cytochrome C is covalently attached through cysteine residues, so we need cysteine there. Cysteine is the one that has the sulfur. We also have iron-sulfur proteins. These contain iron with inorganic sulfur atoms, and with the cysteine residues. Again, cysteine has sulfur in it, so it's very important, that type of protein. They only carry 1 electron. Okay, let's talk about the standard reduction potential. So the standard reduction potential basically says low E, naught goes to high E, naught. So what do I mean by that? So NADH has a very low, this is a low E, naught, and then you're going to flow the electron from NADH to ubiquinol, and then to cytochrome C. H2O has the highest E, naught. So ultimately it's going to go from NADH to ubiquinol to cytochrome C, and then to H2O. Again, H2O is the final electron acceptor. The order of the carriers is NADH, which we have there, so NADH to Q, which is our ubiquinol, actually QH2, then cytochrome B, then to C1, cytochrome C, cytochrome A, cytochrome A3, and then O2 as the final one. We have 4 unique electron carrier complexes, and now we're actually going to go through them in depth. Complex 1 is NADH to ubiquinone. It's also known as NADH dehydrogenase. This has a ton of polypeptide chains. It takes the 2 electrons of NADH, and it will pump out 4 hydrogens to the inner membrane space in the mitochondria. There's also these iron-sulfur centers that are going to be passing that electron to ubiquinone. So what happens here, so we have NADH plus H. This is in the negative area, the matrix, all right? And then it's going to go through complex 1. And then we're going to get NAD. This is oxidized. Oxidized form is NAD. And then we're going to get this QH2. So it's not ubiquinone anymore. This is ubiquinol. And 4 protons are going to go out into the intermembrane space, so plus 4 H pluses. But we put a positive over there, so positive because, again, the inner membrane space is positive, the matrix of the mitochondria is negative. Complex 1 and complex 2, so what's interesting is both of them are kind of starting points. You don't go complex 1 to complex 2. You could go complex 1 to complex 3 to complex 4, or complex 2 to complex 3 to complex 4. Okay, complex number 2 is also succinate dehydrogenase from the TCA cycle. This is the one that took our succinate and it became fumarate when we were studying the citric acid cycle. Again, this is going to take 2 electrons from FADH2 this time. it's not a proton pump. So this one does not pump out any hydrogens into the intermembrane space. The heme B prevents electrons from leaking and the formation of reactive oxygen species. We don't want those ever formed. FADH2 plus Q, again, you're going to have the FAD, so the oxidized form is FAD plus our ubiquinol, and that's going to be the equation for complex 2. Again, complex 2 is not a hydrogen pump or a proton pump. Complex 3, you're actually going to do complex 3 twice. That's because cytochrome C, remember, this cytochrome C only carries 1 electron, but we have 2 from either complex 2 or 2 from complex 1. So we need to actually do complex 3 twice. Cytochrome C also moves in the intermembrane space, which is very interesting. Overall, you are going to be moving 2 electrons from our ubiquinol, and 4 hydrogens are going to be pumped out. The overall equation would be QH2, which is again ubiquinol, plus 2 cytochrome Cs, plus 2 H's. That's on the negative side, remember, in the matrix. And then you go to this ubiquinone. It's not ubiquinol anymore. It's ubiquinone because we took away the hydrogens, plus the 2 cytochrome Cs, which are now reduced. And then you're going to pump out your 4 hydrogens into the intermembrane space with complex 3. Complex 4 will be cytochrome C to the matrix's O2. Remember, oxygen is that last carrier of the electrons. This is going to form water. So complex 4 also pumps out 2 hydrogens. So the overall reaction, let me get to it first, will be cytochrome C plus ubiquinone, and that's going to diffuse between the supercomplexes. This prevents any leakage or formation of reactive oxygen species. Something interesting here, cytochrome C also absorbs visible light. It becomes red. Okay, let's talk about the standard potential charge or change. Let me go down to that one. So the standard reduction potential change is this delta E equals the E of the acceptor minus the E of the donor. And when you're dealing with these equations, usually the more greater the number, the more positive value is going to be the reduced one. So with that in mind, you could think of delta E equaling reduction E minus oxidized E. Okay, so let's do an example of getting the standard free energy change. So, standard free energy change, that will be when we take our, let me see. Okay, so we have this equation, delta G is equal to negative NF delta E. This N is going to be the number of electrons, if I remember correctly. Yep. F is a Faraday constant, and then delta E is going to be acceptor minus donor. So let's put in some numbers. So the delta, or just the E for oxygen, O2 is equal to 0.816 V. And then the E for our NADH is negative 0.32. So NADH is equal to negative 0.32 volts. Okay, so how are we going to first calculate our delta E? So we could do this first equation, the standard potential change. So remember, the more positive or greater the number, that's most likely to be the reduced number in the equation. So we could set up delta E is equal to the E of the reduced minus the E of the oxidized or the donor. Okay, so that means delta E is equal to our number for oxygen is a larger number than the number for the NADH. So we're going to put that number for oxygen first. This one is going to be first because it's, again, the more positive number. So 0.816 minus a negative 0.32. That will get us to this number, 1.14 volts. Okay, so now we plug in this to our delta G equation. NADH carries 2 electrons, so the equation is going to be negative 2 times, in parentheses, is 96.5. That's a given constant. And then times 1.14 V, which was our delta E that we figured out. And our answer is negative 220 kilojoules per mole of NADH. Okay, the proton motive force. This is going to, again, you need a gradient. This is going to involve, again, that idea that we're pumping the protons out to the intermembrane space. They're going to be causing this large amount of positives. And then the negative part of the mitochondria is going to be the mitochondrial matrix. Inhibitors of the electron transport chain would be the ATP synthase inhibitor would be oligomycin and ventricidin. I don't know how to say that, but oligomycin is the one that inhibits ATP synthase. Uncoupling reagents such as DNP and uncoupling protein 1, these are going to basically separate ATP synthase from the electron transport chain. Uncoupling protein 1 is found in brown adipose tissues. It forms proton-conducting pores in the inner mitochondrial membrane. Okay, let's talk about reactive oxygen species, which are generated during oxidative phosphorylation. We have superoxide dismutase as an enzyme. This is going to convert superoxide, which is, let me find this. Superoxide is O2 minus, it looks like. Superoxide dismutase will turn that O2 minus into H2O2 as well as H2O. Oh wait, H2O2, sorry. So superoxide dismutase turns the superoxide into H2O2 as well as O2. Sorry, that's what I was missing. And then we could go further with glutathione peroxidase, another enzyme here. Glutathione peroxidase will take H2O2 and convert it into water. We also have nicotinamide nucleotide transdehydrogenase, as well as glutathione reductase, but those ones weren't highlighted, it looks like. Okay, let's talk about the ATP synthase itself. This is going to be powered by the downhill hydrogens moving back into the matrix. This is coupled with the electron transport chain. Again, if you get rid of one, the whole system won't work unless you've uncoupled them. The overall reaction will be ADP plus P plus just hydrogens. This is going to be in the positive side, and it's going to be ATP plus H2O plus the amount of hydrogens too on the negative side. That's going to be the overall. How are you going to uncouple the oxidative phosphorylation? Well, one way to do so is to just use physical shear or detergents. You could also use DNP. DNP is 2,4-dinitrophenol, and this is going to be a weak acid that carries hydrogens across the membrane. Since it carries the hydrogens across the membrane, this dissipates the protein gradient. It gets rid of the proton gradient. Let's talk about the domains of ATP synthase. There are two of them. There's the integral part, F0. It has this pore. for protons. This is stuck in the membrane of the mitochondria, and that's given by the integral nature of it. The peripheral part of the ATP synthase, this is a thing where if it will catalyze the hydrolysis of ATP if it's isolated. Each beta subunit of ATP synthase can assume 3 different conformations. Mitochondrial F1 will have the alpha, beta, gamma, delta, and epsilon. So I forgot these ones. Each beta subunit has a different conformation. We have beta ADP, beta ATP, T as in Tom, and beta MT as our 3 conformations. The structure of the F0 complex, the one that's in the membrane that has the proton pore, which is an integral protein, the F0, this is only going to have AB to CN. N ranges from 8 to 17 depending on the species, and we have this C ring which will rotate counterclockwise. The C subunits will rotate together as a unit to bring in protons. This is this idea of rotational catalysis. We're going to be causing these protons to flow through F0, and this is going to cause the C ring to rotate and in turn trigger the subunit conformational changes in F1. We also have this binding change in F1. The translocations will cause the force of the proton flowing through to create 1 ATP. That's the binding change. It goes through these 3 different conformational forms. Okay, how transport happens. So we still need to bring some things in. Adenine nucleotide translocase. This is an antiporter that will be moving ADP into the matrix while pushing ATP out. And then we also have phosphate translocase. This one's very important. This kind of gives us the answer why we need 4 hydrogens instead of 3 hydrogens for 1 ATP. So phosphate translocase, we need this hydrogen to move in H2PO4. Remember, we had that inorganic phosphate, so that's going to be a problem to bring it in. However, we could couple it with a hydrogen and this phosphate translocase, and we could move H2PO4 into the matrix. So, in reality, you actually need 4 protons for 1 ATP. So, an overall equation that was given is X ADP plus X phosphates plus 1/2 O2s plus H plus NADH will give you X ATP plus H2O plus NAD. Let's talk about the phosphate oxygen or P2E ratio. This is the value of X in that equation here, this equation here. There's no theoretical requirement for phosphate over oxygen to be integral. The last part of this, or second to last I should say, we're going to be talking about how we got those numbers, the 2.5 and the 1.5. So, 2.5 ATP, remember that's with NADH, right? 2.5 is when you're entering complex 1. However, 1.5 ATP from FAD, that's when you're entering ubiquinone instead. So, the path for NADH is complex 1 to complex 3 to complex 4. Remember, complex 1 pumps out 4 hydrogens. Complex 3 pumps out another 4 hydrogens. Complex 4 only pumps out 2 hydrogens. So if we do 4 plus 4 plus 2, we get to 10 hydrogens. FADH. Remember, with FADH2, we're starting at complex 2, not a protein pump, so nothing there. Complex 3 pumps out 4 hydrogens. Complex 4 pumps out 2 hydrogens. for a grand total of 8 hydrogens if we started with FADH2. Okay, 4 ATP, 4 hydrogens per ATP, right? We established that earlier. So we're going to take 10 divided by 4 hydrogens, and that's going to give us 2.5 for NADH. That's where we get that number. And then 6 hydrogens, which are pumped out when we start at complex 2, divided by the 4 that are needed per ATP, that's going to give us 1.5. That's where we get that number. Okay, let's talk about regulation as our last bit here. Regulation. So usually the acceptor control of respiration is just the ADP level. We could look at the mass action ratio, which is ATP over the concentration of ADP. Make that better. And the concentration of phosphate. Usually this ratio is very high. When it's lowered, this increases the rate of respiration. When it's lowered, this means more ADP is available. Let's also talk about inhibitory protein. IF1, this is going to bind 2 ATP synthases together to inhibit enzyme activity in both of them. It's active in its dimeric form as a dimer at pHs less than 6.5. Where do we find this very acidic pH? We could see this in the muscles when there's a ton of lactic acid. In hypoxic or oxygen-deprived cells, IF1 binds and prevents a drastic drop in ATP. And then again, ATP-producing pathways are coordinately regulated. ATP and ADP concentrations control the rates of ATP synthesis, of course, but also the electron transport chain, citric acid cycle, pyruvate reactions, as well as glycolysis. All right, those were all the notes on Chapter 19 about oxidative phosphorylation. Please stay tuned for the ones on photosynthesis. Take care and have a good one. Do something nice for someone.